Thank you.
Hello, everyone. Welcome to the MIF++ seminar. It's a great pleasure to introduce Professor Elspeth Garman from the University of Oxford, who will talk about two different challenges in biological structure determination: radiation damage and metal identification. Over to you,
Thank you.
Elspeth, please.
Thank you very much for the introduction and also for giving me the chance to talk to you. And thank you for coming along because it's a grey winter day here and probably you want to be by the fire. So yes, the two challenges. This is a sort of follow-on from Alex Vlodar's talk about the infelicities that he's discovered in the protein data bank. But this addresses two very specific problems. So I'm going to first of all just introduce the phenomenon and remind you what we do in structural biology to find three-dimensional structures of biologically interesting molecules. So if we look at the pipeline, and it's a very linear pipeline in that you can't possibly do any of these steps unless you've done the first step. So you need to get pure protein. You need to pray to the gods that you believe in and try and grow a crystal, which is still a black art. Collect data by irradiating the crystals with X-rays, usually now at a synchrotron, also in-house sometimes with copper K alpha radiation. Then determine the structure. This step has suddenly become a lot easier because of AlphaFold 2, and I know you've had some talks about that too. And then we get the atomic coordinates. Now, the step between here and here involves taking the spots on the diffraction pattern, finding the square root of the intensities, or a quantity proportional to the square root of the intensity of those spots, the so-called structure factor. And then if we know the phase, which we can now get from AlphaFold model of the protein, I'd like to emphasize that these AlphaFold models are hypotheses, they are not structures. And although everyone's saying crystallography is dead, my argument is that we still need to confirm them, and some of them are in fact incorrect. So there's still a place for crystallography. So if we look at how it's developed in the last 50 years, 70 years, we saw Dorothy Hodgkin solve the structure of penicillin, which was 25 non-hydrogen atoms. And now in X-ray crystallography, we don't see the hydrogen atoms. You need to do neutron crystallography for that. Because the cross-section for X-rays for hydrogen atoms is very, very low, as we'll see in a minute. And then she solved vitamin B12. That was 80 non-hydrogen atoms. And then insulin, which was 30 years' work, 829 non-hydrogen atoms. But just to put you in the picture of what's possible these days, the 2009 Nobel Prize went to the people who did the structure of the ribosome, which was Venki Ramakrishnan, Ada Yonath, and Tom Steitz. And that had 293,658 non-hydrogen atoms. So we can really do X-ray crystallography now on the most enormous structures. What do we actually do? Well, probably many of you know this already, but we can have an X-ray source, as I said, synchrotron or a lab source. We have some conditioning optics for the X-ray beam. There are no lenses which will allow you to focus an X-ray beam. Well, not simple lenses anyway, no optical material. But some very fancy multi-layer lenses allow you to actually, relying on Bragg's law, allow you to focus the beam onto the crystal. And in this picture, this diagram here, the picture is the crystal is incredibly out of proportion because it wouldn't even be 1 pixel of the picture. And then commonly we have 100 Kelvin nitrogen gas coming onto the crystal to cool it down in order to slow down the rate of radiation damage, which I'm now going to talk about a little bit. So we can use 5-micron crystals now at the synchrotron because the beams have such a high flux density, and we need about 50-micron crystals at home. So these are small and we have to deal with them under the microscope. We then, as I say, go to the synchrotron. We cryo-cool the crystals by looping them up like you blow bubbles in the bath, if you still do, or for your family, using surface tension forces, and plunge them into liquid nitrogen, having cryoprotected them. But that's a whole nother subject, which I won't go into today. We get a diffraction pattern, and then using Bragg's law and the Fourier transform, if we've got some phases, either from an experiment of doing the experiment at various incident wavelengths, X-ray wavelengths, or from an AlphaFold 2 model, where we've stolen the phases as a starting point, we get an electron density map which is the green here. And what we have to do is get the yellow model and get the model to fit the data, which is the green. And then we refine that in an iterative procedure. And this is the best one I ever got to make you believe in atoms. So usually this is what small molecule structures look like, but in this case, this is quite a large protein from salmonella food poisoning that we solved, because the crystals were beautiful and diffracted to a very, very high resolution, which means that the Bragg's law was obeyed way out at the edges of the detector. So the problem with this whole technique of structural biology structure determination is radiation damage. So I'm just going to take you through what we observed that. And the first thing is to realize that protein crystals are in fact actually somewhere between solid and liquid. They're not pure solid and they're not pure liquid. So you can liken it to synchronized swimmers in a pool. And what you notice about these swimmers is that some of them have got bigger mouths than others, and some of them have got their fingers in slightly different places. And this is very representative of how a protein crystal will have different conformations of the protein, of the loops, the flexible loops. But what we end up with in our electron density map at the end of our structure determination is actually a space and a time average of all of the protein molecules, i.e. the swimmers. But what you notice is that they're in water. And our crystals, our protein molecules are indeed, they don't pack neatly together because they're such funny shapes. There's spaces between them, which is full of the buffers and the water that we grow the crystals in. So this means we have to treat them with great respect. And if we take them out into the air, then the top layers dehydrate and all these molecules become disordered and they're not lined up anymore. They're not ordered. So then we don't get good diffraction. So the whole treatment of the crystals has to be quite careful. If we put at room temperature a crystal into a third generation synchrotron beam, such as at the microfocus beamline at Diamond I-24, these crystals are in a tray, so they haven't been taken out of their growth tray, the plastic tray. You can see the crystals themselves are about 50 microns in square. If we take two images here, we then move the tray and take two images here, while the bit that we've just irradiated breaks up. Then the second bit breaks up, and with persistence we can get from about 50 crystals enough images to actually get the structure of this bovine enterovirus. And you can see here the radiation damage to this crystal, which apparently was about 100 microns. You can see that they actually managed to etch their beamline number onto the crystal. Now, this is not diffraction, and it's not what we want to happen. So this is a sign of the radiation damage. But what we see in the diffraction pattern as we irradiate the crystal is that at the beginning, with Bragg's law, as I said, the high resolution information is all in the reflections out here. The low resolution information is in the middle here. And as we irradiate the crystal at room temperature extremely quickly, we see that all the high resolution ones have gone. They've disappeared, so that the disorder means that we can only now see the envelope shape of the protein. You can see that everything out here has gone. This is about 3.5 angstroms is this solvent ring here. This ring is due to the scattering from the liquid in between the protein molecules. So we then have incomplete data, and it's low resolution data. So what's happening here is that the diffraction, which is elastic scattering, doesn't damage the crystal because it doesn't leave any energy in there. But the absorption components, which is Compton scattering, where an X-ray comes in and goes out with slightly less energy, ejecting an outer shell electron usually, and the energy will be lower coming out and the wavelength longer. That's about 8% of what happens at 12.4 incident energy, which is a common energy that we use. What really kills this is the photoelectric effect, where we eject an inner shell electron, and then that electron, we absorb the X-ray completely. We eject an inner shell electron, which has enough energy to ionize 500 further atoms. Now, these can spread. So what you see here is a direct ionization and absorption event on the protein and one on the solvent, and as I say, 500 electrons can then be produced by this energetic electron from the photoelectric effect and spread through the structure, spreading the damage. And what we would like to do is immobilize all these secondaries, because we can't do anything about the red ones, which are the primary events. That's a fact of physics. So what can we do? Well, we can cool the crystal, as I already mentioned, to 100 Kelvin. And it turns out that if we do that, we get 70 times on average more data at 100 Kelvin, holding it in a stream of gaseous nitrogen, than we get at room temperature. And if we look at the intensity of the reflection staying up as we collect data, rotate the crystal in the beam to sample reciprocal space in 3 dimensions all the way around, we see that this at 120 Kelvin, the intensity stays up, whereas at 300 it drops. And if we look at the resolution of the data, so the detail of the biological molecule that we can get at 300 K, this starts to go, this is a quality indicator about how well the internal data agree. If I actually detect the same reflection twice in the data set, how well it agrees. So the bigger the number, the worse the quality of the data. And you see that what cooling allows me to do is give me enough time to measure the high resolution data. It doesn't make the crystal diffract any better at the beginning, but it does give me time before the disorder sets in to actually collect the data. So this has had a huge impact on the structural biology field. And if we look at the protein data bank, which I think you're all familiar with, which now has 228,000 structures in it, about 200,000 of those are X-ray structures, slightly more than. The blue here is cryo-cooled structures, and the red is room temperature. And it's only in the last 5 years when it's been compulsory to enter the temperature of the collection. Now you might say, but my body isn't at 100 Kelvin. Are these structures really what the proteins look like in 3 dimensions? And the answer is that the predictive power of them is very great. But if you want to spend $1 billion developing a inhibitor or a drug for a protein, then it's very good to also have the structure at room temperature, so you know what the water structure is and what the disordered loops are doing, because what cooling it does is actually damp out all the dynamic disorder on the structure. So as you can see, it completely dominates the way people do structural biology now is the cooling. However, even when you cool the crystal, you start to see pretty catastrophic things happening. So this is a bird flu neuraminidase crystal from a noddy tern in the Great Barrier Reef, neuraminidase from flu virus, and you can see the marks that the beam have left. This was an iron-containing protein up here, and the beams left black marks on it. And then when you warm up the crystal, you can see that the radicals produced by the X-ray beam absorption have caused gas to come off, and the whole crystal breaks up just completely into smithereens. And we see spectral changes as well. We also see the dimming of the high resolution reflections again, and some other features as well. And if we look, we first observed these at a flux density of 10 to the 12 photons per second into 100 micron square slits. But if we look now at the equivalent flux densities available, I-24 on diamond light source, that's 700 times what we observed these effects in 1999 and 2000 first coming. So we've got a serious problem with the flux densities of these synchrotrons, even at cryo temperatures. Now the question is, can we actually quantitate what's going on so that we can predict how long the crystal will last in the beam, and then change our data collection strategy so that we get the data that we want? And if we want to compare experiments from different synchrotrons, different beamlines, different days, and different proteins, what we need is something along the x-axis, which isn't the number of images or just the time, because we can't compare that. So we use this rather unsatisfactory, but it's all we've got, quantity called dose. And now a dose is a physics estimator of how much energy is absorbed by the sample. I can't actually measure the dose, I can only estimate it. It's the energy absorbed per unit mass, joules per kilogram. Crystals commonly weigh about a microgram or even a nanogram, so this is quite a strange unit, but it's measured in gray, uppercase G, lowercase y. And this is a fundamental metric. and we've tried to get the field to actually plot radiation damage indicators against this metric. The problem is you need to be able to estimate it. And so we also wrote some software called RADDOS, which is housed in Sweden at a website, and anyone can get on this website and calculate what their beam is doing to their crystal in terms of dose. And we've just published a fourth paper where I'll talk a bit more about this in a minute. But you can estimate the dose, as I say, but you can't measure it. So we see the intensity of subsequent data sets going down like this. It's actually exponential, but at the beginning it's very, very linear, and we gradually see the reflections disappearing. We also see a very strange effect, which is that the unit cell, the translational repeat unit of our crystal, appears to expand. And we think this is gas which collects at dislocations and then makes it look as if there's an expansion, because actually the protein shrinks a little bit when you cool it. And this is linear, but then it's not reproducible between crystals of the same protein on the same day with the wind in the same direction. So unfortunately, we can't use it as a metric to say when your unit cell expansion is more than 0.4%, throw the crystal away, because it's so variable, and it depends on the number of, seems to depend on the number of dislocations and imperfections in the crystal. There's a second class of damage, though. So everything I've shown you so far is in reciprocal space, in diffraction space. A much more worrying and less obvious at the beginning type of damage is specific structural damage, where we affect particular amino acids in a very, very well-defined pecking order. And one of the pioneers of actually characterizing this is my dear colleague Raymond Ravelli, who sadly died last year of pancreatic cancer, and we all miss him terrifically. But this is a shout out for his very good work. This is one of his plots of lysozyme, which is a protein in tears. This is hen egg white lysozyme, and it's a difference map. What that means is it's the fourth data set, and with the structure factors, the square root of the intensities of the reflections have been subtracted from the first data set to see what the differences are. And red means there are things missing in the fourth data set, and green means there are extra things in the fourth data set. So if we look at the disulfide bonds here, we see that both the sulfurs have moved apart. Here's a solvent accessible one, and up here, the fourth one here, the sulfurs have actually moved to new positions, which is what the green is. So the first thing we observe is the breakage of disulfide bonds, which requires 2 electrons to break it. Now, it turns out that these electrons that I mentioned are actually mobile and can move along the amino acid backbone and look for the most electroaphilic site, and then they will facilitate the breakage of the disulfide bonds. Now, the second thing that happens in the second order is that aspartates and glutamates become decarboxylated, and that's what you're seeing here. And we have actually observed carbon dioxide coming off our protein, which then got trapped, well, let me show you these first. So the first thing, of course, that happens, which I haven't mentioned, is a reduction in metallocenters, then the breakage of disulfide bonds, and the metallocenter reduction happens extremely fast, and that we can observe that through doing microspectrophotometry on our crystals as we actually irradiate them with X-rays, and we can observe the reduction of the metal centers through that. The disulfide bonds, then the asp and glue decarboxylation, and then finally, well, not finally, but next, we see the cleavage of the sulfur carbon bond in the methionine. Now, what's very different, also, sorry, if you've got any covalent bonds to heavier atoms like bromine, iodine, and mercury, you see them breaking as well. So what's interesting here is that if it was just in the order of the cross-section of the atoms to the X-rays, then we would see the breakage of the disulfide bonds, then the sulfur carbon bond breaking, but we don't. We see this, and that means that there's definitely something mobile even at 100 Kelvin. And this is the electrons which can quantum mechanically tunnel around the structure, and also a bit of thermal energy still at 100 Kelvin. Now, when I learned about quantum mechanical tunneling in my physics degree many years ago, I thought this is never going to be any use to me, but it turns out that this is exactly what's happening here. And up here, you can see exactly what I meant about the breakage of that disulfide bond and the two new positions that the sulfurs take up. So here is the carbon dioxide, which we actually trapped coming off. I mean, it got trapped and we could build it in the electron density. So the purple is the electron density from the experiment. The yellow and blue and red is our model that we have fitted to that, built into the electron density. And then this is the carbon dioxide, just to show you that this is not a figment of our imagination. The second thing, as well as the structural damage that happens at cryo temperatures, is that the atomic B factors, that is the uncertainty in position of the atoms, increases with dose, so increases as you collect data. Now, this is going to be very important in a minute when I explain the new metric that we've come up with to look at radiation damage in the protein data bank individual structures, where we don't have any dose information or any irradiation information. If we look at what happens comparing cryo temperature and room temperature for the global damage, that's the in reciprocal space, and the specific damage, which is the green here at cryo, with the room temperature, we can see that at cryo we get much less damage, much slower damage in reciprocal space, but that the specific damage happens much sooner than the diffraction damage, but that at room temperature, they happen concurrently. So if we blow up this little area here, which is the room temperature global damage and the room temperature specific damage, which is in this box up here, you can see that the scaling B factors in reciprocal space start going down, but there isn't nearly as much gap between the structural damage, the atomic B factors going up at room temperature. So that's because the lattice disintegrates much, much faster, because the radicals can move around, they have temperature energy. So just in summary of what I've said about the pathology of the damage, in global radiation damage in reciprocal space, we've got decreased intensities, unit cell expansion, scaling B factors for each data set goes up, and then we get increase in the disorder of the protein. And we can see that because the spots get a bit more smeary, because the Bragg condition is actually obeyed in different orientations of this one compared to this one. So we'll get more smeary spots for this case. Then in specific radiation damage in real space, we see chemical changes in reduction of metal ions, the disulfide bonds, the glutamate aspartate decarboxylation. And we did think that we saw side chain, but we did think that the OHs came off from tyrosines, because we always saw difference density around this OH here. But in fact, we did a special study of all the cases in the protein data bank and showed that this is the whole tyrosine moving, because radiation chemists tell us this is impossible for this to come off. It's energetically not possible. And so that's been debunked now as one of the phenomena that OH comes off. I already mentioned what's happening here with elastic scattering, Compton scattering, and photoelectric absorption. It's this last thing which really kills us. If we look at the cross sections, I mentioned that hydrogen was too small for us to see in X-ray crystallography, but carbon, nitrogen, oxygen, these are in proportion to their X-ray cross section at incident energy of 13 keV X-ray. And they're in nuclear physics units, which are in barns, which is a sort of joke. And yeah, you might not think it's very funny. But if we then make a selenium derivative of our crystal in order to get the phasing information at different incident wavelengths and get the phases, we see that it dies in the X-ray beam twice as fast. And that's because that's the cross-section of selenium if we draw it in proportion. So you can see that what's actually in the crystal really matters as how fast it will radiation damage. What's causing us trouble here are OH radicals, which we think are mobile above 110 Kelvin, which is why we try to keep the crystal below 110 Kelvin, and the electrons, which I already mentioned, we think can quantum mechanically tunnel around the structure. So why do we care about this? Why am I telling you all about it? Well, there's three reasons. One is that we don't get all the data we need because the diffraction pattern fades away. The second is that if we are going to do a multi-wavelength anomalous dispersion experiment to get our phases, then we've got some things that are not the same at the end of the experiment as they were at the beginning of the experiment. We've got this cell expansion, we've got the structural changes due to the breakage of the bonds, and something I didn't mention, which is that it appears that the cell is redefined by the data software, and it looks like the molecule is actually moving in the unit cell. In fact, it isn't. These effects completely wash out the signal that we need and stop us solving the structures. As I say, now with AlphaFold 2, that's not as serious as it was even 3 years ago. So the main reason we care, though, is that if these effects can affect what we think about our biological results. So metalloproteins, as I've mentioned, are often photoreduced. We get decarboxyl glutamate, and that is sometimes part of the protein mechanism. So it's indistinguishable from radiation damage. And there's a group in Japan that have spent many years untangling the structural intermediates of bacteriorhodopsin, which is a very important membrane protein for our optical system, and trying to untangle that from intermediates induced by the X-ray beam. So there's a dose limit postulate due to Richard Henderson in 1990 that there's a maximum amount of energy that you can absorb in a biological sample before it will be compromised. And he calculated from what he'd observed in electron diffraction that this would be about 20 megagray to take the intensity of the pattern down to half of what it had been at the beginning of the experiment if this sample was held at 77 Kelvin. And he made it very clear that the crystals might not reach that limit, but they couldn't be expected to go beyond it. So we measured this limit and we actually got 43 megagray, but at that dose, there was so much damage to specific structural amino acids that we actually said it's not safe to take it down to half of the intensity, only to 0.7 at the absolute most, and that was 30 megagray, and that's what's known as the experimental dose limit. Now, I went to see Richard Henderson before we actually tried to publish our experiment where we'd measured it. And he said, "Oh, what do you get? What do you get for the limit?" And I said, "Oh, I'm really sorry, Richard. We got 43 megagray, not 20." And he said, "Oh, thank goodness. I thought I'd be a factor of 10 out." So that was fine, and it was calculated. So to do this, you need to be able to calculate the dose, and just to orientate you with doses, a typical protein crystal which hasn't got any metal in it, so it's not terrifically absorbing, only organic elements and sulfur, will absorb about a megagray a second. And this little hamster here, if he gets 2 to 10 gray, he will die in 10 to 30 days. And a human brain tumor gets 60 gray absolute maximum in 30 doses of 2 gray a day. So you can see that these protein crystals are incredible what they'll withstand in terms of absorbed radiation. 1 megagray of absorbed dose rather, will give 1 ionization for every 20 amino acids in the protein structure. That's for a 400 amino acid protein. So at room temperature, you reach this limit in about, the limit is much lower, about 70 times lower, and you reach that 0.4 megagray in about 0.4 of a second, and at a modern synchrotron. And for 30 megagray, you'll reach it in about 30 seconds. But for the new sources, the 4th generation sources, it's less than 1 second that you have to collect your data set, which means that you've got to be really careful with what you're doing. At free electron lasers, you get damage before destruction is they always say diffraction before destruction, but in fact what has been shown now is that if you have a pulse length which is longer than about 20 femtoseconds, you can actually see specific damage to the structure. So you've got less than 80 femtoseconds to reach the limit. This is what you need in order to calculate the dose, and I won't go into this, only that it's available as raddo.se, but you do need to be able to characterize both your crystal, here we are, and your beam. So you need to know the flux density, the size and profile of the beam before you can calculate the absorbed energy. This is now pretty sophisticated code written by my group over the last 10 years in Java. My original version was in Fortran, but I can't write Java, sadly. But it's being used at synchrotrons, quite a lot of synchrotrons all around the world, to actually allow experimenters to limit the dose that they allow their crystals to absorb. There's a GUI now written by my ex-student Josh Dickerson, who's now in Berkeley, and it allows you to actually map the X-ray dose, the dose that you're absorbing for different strategies. So this is the fried egg, and the middle here is 30 megagray, and then it goes out 20 megagray, 10 megagray absorbed dose. This is to take 360 degrees of data, so rotate the crystal completely. And if I was live, I would be doing this with my body to show you how it works. But I call this the Newton's cradle. If your crystal was long like this rectangular one here, then you could move it and do 10 degrees at different places, or you could move it and translate it at the same time. But you can see that you can model with the software different data collection strategies in order to optimize the use of your crystal volume. This is a very alarming plot that we made last year from all the data from the new synchrotrons and the existing ones. Up here is the time you've got before you reach the cryo dose limit, which is 30 megagray, as I mentioned, and the time you've got at room temperature on this axis to reach the 0.4 megagray. And here's the dose rate that these beams deliver. And you can see that these are all different beam lines at synchrotrons around the world. And the latest one here, you've got less than just over a millisecond before you, that's for your 100K. So this has resulted in people using serial crystallography, as it's called, where you just get one shot per crystal because the radiation damage is so high that that's all you can manage to get. I won't go into that, but we have now, just to say that we've got Raddose 3D for free electron lasers, for SACs, small angle scattering experiments, small molecule crystallography, powder diffraction, and X-ray and XPS as well. And we've just released Raddose electron diffraction as well, because what we would like to do is get electron microscopists to stop talking in electrons per square angstrom, because there are different energy electron microscopes, 100 kV, 200 kV, etc., 300 kV, and of course they cause different absorption depending on the energy of the electron. So we would really like them to start talking in grey instead of in electrons per angstrom. But this is going to be an uphill struggle, but we're trying. And now we've released the software so that they can actually do that conversion quite easily. And you can see that this is the efficiency coefficient, which is the inelastic to the intensity of the pattern per megagray, and which is the so-called information coefficient. Sorry, not the intensity coefficient. And then this is the thickness of the sample here. So you can see that it matters, A with the thickness and B with the energy of the electrons. Right, so that's I'm going to move on to talking about what the abstract said, which was the radiation damage in the PDB. So this, we've now talked quite a lot about the data collection and the effects on the structure determination, but this is now the atomic coordinates. So what I mentioned was that radiation damage causes the atomic B factor to go up as you irradiate it. So what we have in the protein data bank for every atom is XYZ position, the B factor, which is its locus of occupation, which is usually isotropic, so a sphere, even though they would be better as anisotropic, but we usually don't have enough data to fit anisotropic B factors. And then the occupancy, which is usually 1 for amino acids, and it might be less for ligands and so on. So can we take an isolated PDB file, any random one, 100K one, from the protein data bank and calculate how damaged it is? What we used for this was the 30 radiation damage series in the protein data bank, where we tested our metric to see if the signal was good, and it was good. And I won't go into the detail of how we validated it, but what did we use? There is an issue here with the B factor. So if I'm an atom near the surface of the protein, I have a lot more degrees of freedom where I can move around, and I'll have a bigger B factor because the packing density is lower than I will if I'm stuck in the middle of the protein, where I can't move around. So here we see that the packing density here is very high because I'm completely surrounded in other amino acids, whereas here it's very low because I'm near the solvent. The B factor here is low, but at the surface it's high. So the question is, can I deconvolute the packing density from the B factor number to get rid of this dependence on how near the surface the protein is, or how the amino acid is, how easy it is for it to move around? And the answer is yes, and that's this B damage metric that we developed some years ago. And basically what we did was say, what's the average B factor of atoms that are in a similar packing density to our atom of interest, and divide it out and see what was left. So it's corrected for the packing density. And then we'd take the average B factor of all the atoms with a similar packing density, divide it into it. So this allows us to give a B damage value to every atom in a structure, every non-hydrogen atom in a structure, which is what we've done here for acetylcholinesterase, which is an important protein that's been studied by Martin's White group over the years. And you can see here, I mentioned that disulfide bonds are very susceptible to radiation damage. And if you look here, the B damage value is above 1. Here it's 1.62. This one breaks before this one, and this one breaks at about the same time. So the B damage metric allows you to then say, what were the hot spots of my structure? So this is a structure all on its own. I don't have a series, a radiation damage series for it. So I've just got one data set. And then I can go and look at the electron density at those places and do a difference map between the model and the data, and see if there's anything missing in the data. And you see here, it's exactly what I've done for this example, acetylcholinesterase. And once you've identified high B damage values, you go and have a look, and you can inspect the electron density and see that there's difference density here. And this one was damaged less. This one was the highest one. So now the question is, if we've got values for all of these atoms on this structure, can we use this to say, is there a single number I can give this so that I can see if it's damaged? And the answer is yes. And I had a very, very fantastic student called Catherine Shelley, who's now with David Baker's group in Seattle, who I had 6 ideas about how to do this, and none of them worked. And this was her idea, and it did work. So what we did was we took the median of all of the B damage values in the whole structure, and then we plotted the B damage values for the asps and glues, the oxygens for them. And we looked at ones that were damaged and ones that were not damaged. So here is a low dose. This is from a series, a radiation damage series. This is a low dose asp and glue oxygen plot, and this is a high dose one. And it turned out if we took the median here and then took the area above the median and divided by the area below the median, we got a number that we relied on, which we could rely on. We tested it on the 30 different radiation damage series in the protein data bank. And this metric we call Bnet. And here is an example where this is low B damage is low blue and then up to red, and you can see there's some red bits on here, but this has come out with an index of 1.4. So is that a good or a bad index? Well, it turned out that anything with an index above 3, a Bnet above 3, had serious damage. So here are 2 examples of 78 and 66, so very high Bnet values, and the aspartates and glutamates were basically not there you can see the difference map there. So the blue is the data, and then this is the red is the difference between the model and the data. And you see the model has the glutamate in, and the data there's just no density there at all. So we published the worst 10 of these in a Nature Comms paper. This one was just a terrible structure, but you can see that all of these have something wrong with them in terms of radiation damage. And it allows you, if you're using a structure, to just run the software, which is called RabDam, and give it an index and see whether you can rely on it or whether you should look at the difference density. So just in summary, we can detect potential sites of specific damage using this Bnet, which is derived from the B damage values. There's a CCP4 program called RabDam now, which allows you to do that very easily. And you can open up the entire PDB for a large-scale statistical analysis of specific radiation damage. And if I had any more students, that's exactly what we'd do. But I've retired now, so I don't. But Catherine in Seattle, if she gets time, we'll have a look at this. So at the moment, the radiation damage is overlooked really when you assess the quality of structures in the PDB. And what we would really like to do is to get our metric added to these metrics that you get whenever you put a structure up, which give you the quality. What you want is everything to be at the blue end here, and you can see that this one's a terrible one because everything's red here. Whereas, and most of these don't show anything really bad in the red. And so we would like to get our metric put in here, and we're working at that. It has to fulfill some software requirements, which we're trying to fulfill at the moment. Right, so I'm going to spend the last 10 minutes telling you about metals. So now we've got the fact that we have to be slightly suspicious about radiation damage in the protein data bank, but we have a tool we can use, which is Bnet, to actually see how bad it is, and then whether we have to worry about it or not. So now I'm going to tell you about the metals in the protein data bank. Now, the metals, when you get to the end of your refinement of a structure, can be quite hard to identify, and we use circumstantial evidence. So here's an example of a mystery blob that I'm left with by doing a difference map between my data and my model, and I haven't got this in my model. So it's in my data, but not in my model. So what is it? Well, is it a metal? Yes, it might be a metal. And in order to satisfy my refinement protocol, I'll put a metal in there and just up its atomic number until this difference density disappears. But that's could be wrong because it might be a partially occupied metal site. So it would disappear when I have a metal that's smaller than it really should be. And this has happened over and over again, and people have said, oh, yeah, well, I think it's zinc, so let's put zinc in. Or there's another protein like this in the data bank, and that's got zinc in it, so mine must be zinc. Now, the problem is that the metals are often very important for the function of an enzyme especially. And the other problem is that during our production, our overexpression of proteins in E. coli, they can easily pick up metals along the way that are not what they have in nature. So we've got two problems here. One is that we don't identify the metals very accurately, and the second is it might, even if we do identify it accurately, it might not be the one that's really in nature that matters for this enzyme. So this is a 2014 analysis by Zeng et al. Of the protein data bank. And you can see this is the number of structures, and this is year of publication along here. And you can see that there's a sort of constant number of structures, proportion of structures, I should say, with problematic metal sites, with plausible metal sites, and with no metal sites. And this was just a software tool that they used to get this plot. But as of when they did the study, there were 141 structures in the protein data bank. Well, I updated that this morning, and that was 228,000 in the data bank. So it's gone up by 80,000 since then. But they said 38% have one or more metal ligands. So 38% of today's number means that 86,000 structures in the protein data bank have a metal in. So the question is, how many of those are the right metal and how many of them are the wrong metal? So how can we find out? Well, we can use micro-proton induced X-ray emission or particle induced X-ray emission. So the way this works is that you bombard your atom in this case, part of our protein, with protons, which is the red ones coming in here, knocking out an inner shell electron, here's one, and then an outer shell electron comes down, giving out an X-ray, and that X-ray is exquisitely tuned to the element. So it's going to be a different energy X-ray for every single element in the periodic table. And that means if we could have a really good X-ray detector and detect the energy of these X-rays, rather than just there's an X-ray, yes or no, then we could perhaps see what elements we've got in our sample. And that's exactly what we do. But we also collect the backscattered protons, which are scattering from the nucleus here, because that enables us to characterize the gross matrix composition of our sample and the thickness of it. And we need that so that we can correct for any absorption of the X-rays, because they come out at different energies. They're going to be differentially absorbed by the sample. So it's for analyzing proteins, this is a really almost a perfect technique, because the metal concentration we have is typically one atom or one molecule in 10 to 100 kilodaltons, i.e. parts per million. We've usually got only a small amount of either crystals or solution, so microliters of solution. The absolute concentrations of proteins always have large errors on them, but the absolute magic thing about this technique is that we can use the sulfur content of the proteins, which is the methionines and cysteines. We know the sequence of them. We can count those up and use it as an internal standard. Or if we've got phosphorus in DNA and RNA, we can use that as an internal standard to get the stoichiometry of the metal atoms to the protein atoms. Now, this means that we detect the sulfur peak and we can just count them up, and it's a simple ratio. So how do we do this? We use a 2 MV Tandotron accelerator at the Ion Beam Centre in Surrey University in Guildford. We have a scanning system. Here's the producing the protons here, coming down here, coming down the beamline, a focusing system here with 3 quadrupole magnets, and then a target chamber, evacuated target chamber here with the sample in. So the protein sample is on a little film here, a plastic film in a vacuum. We've got 2 to 3 MeV proton beam coming down, and then we detect the backscattered protons within the chamber, and the silicon lithium drifted silicon detector detects the X-rays. So what does the X-ray spectrum look like? Well, it's down the bottom here. It's a logarithmic scale. We detect everything above magnesium. We can detect magnesium and sodium, but only if we take the filter off the detector, which might damage it. So we prefer not to do that. But you can see the sulfur peak here, which is due to the methionines and the sulfurs in the protein. And there's the zinc, which this protein actually contained. It was a selenium derivative, and it was in a bromide, a potassium bromine buffer. So we can see all these peaks here. The red is the fit to the data. And I mentioned differential absorption. Here's the incident proton beam. So obviously the lower energy X-rays get more self-absorbed in the target than the higher energy ones. So we need to know the thickness of the target, which we can get from the backscattered proton spectrum. So this is a wonderful system developed mainly by Jeff Grime in Surrey to make the software talk to the beamline, etc. And we've now done over 400 proteins measuring the metal content over the last 35 years. So it's a very simple calculation indeed to get the number of atoms of interest. So this might be zinc. We take the concentration of the zinc that we've seen in the spectrum over the concentration of the sulfur, correct for their masses, and then multiply by the number of sulfurs that we've counted up in the sequence, and we can get stoichiometric ratios to within 6 to 15%. So we might have 1 iron atom in a protein. So it's a really good system. So what does this let us do? Well, our calibration protein is cytochrome 550 from Paracoccus. It's got 1 iron per protein molecule. And so our result for them the last time we did this was that we had 0.97 of an ion per protein molecule with an 18% error. It's a 15 kilodalton protein, 135 amino acids with one ion atom and 6 sulfurs. I used 0.3 microliters of protein at 1.35 mg per mil. So you can work out the mass of the protein that we've actually got in our sample is 0.41 of a microgram. There's 5 nanograms of sulfur in there, and there's 0.15 nanograms of iron that we've managed to detect. So we scan the beam in X and Y so that we can get an elemental map of the protein drop by setting a software window around the sulfur peak or the chlorine peak, or in this case phosphorus and iron peak. And then it's like a contour map when you go hiking. We can tell then where we should actually collect point spectra to get better statistics. So you can see here that we collected data on 3 places around here, and the proteins nearly always dry in a ring like this, the liquid proteins. Just one example until I tell you the bad news from the PDB, is this ferric uptake regulator, which was thought to be a binding iron, and therefore the mechanism had been worked out for it to be a ferric uptake regulator from Mycobacterium tuberculosis. But there was something that didn't tie up with the EXAFS data. And so Emke Pohl from Hamburg, now at Durham, contacted me and said, Elspeth, could you look at this? And I rang him up and I said, do you want the good news or the bad news? You've got no iron at all in there. 10 to the minus 5 ions per protein molecule is our lower detection limit, but you've got 3 beautiful zincs in there. And that completely changed what they thought this protein did. So that's just one biological example of how these PIXI measurements can change what the function might be of the protein. What we've done recently is made this a high throughput technique by using a non-contact printer to actually print the proteins in 60 micron size drops, 200 microns apart. And then Jeff has programmed up the computer so that it can collect these data overnight by scanning, moving the stage to the right place for the beam to hit these various samples, so that our throughput has vastly increased from 11 in a day to being able to do 50 overnight and use the beam 24 hours. So what have we done with this? Well, we've actually now measured up 100 samples from the protein data bank of known metalloproteins, where their structure is in the protein data bank with a metal in it. And very alarmingly, we found that the first set was 32 samples. That was the total number. 8 of them had completely different metal from the protein data bank deposited one. 9 of them had additional metals and 13 were correct. And then 2 of them had insufficient data. So we were a bit horrified by this. So we did another set of 28 proteins, and this time 54% had metals inconsistent with a PDB entry. And then we did a third and fourth set of 28 proteins each with a very similar result. Now, 38% of the protein data bank, as I mentioned, 86,000 structures are probably have metal ligands in. If 30% of them are wrong, that's 26,000 of them which may have the wrong metal on in the PDB. So the question is, can we correct them? And I'm just going to show you one example of how we did of this. This was a phosphatase, which means it chops phosphorus, it dephosphorylates other proteins or RNA even, or takes phosphorus off it. This was the difference density here when we did a difference map of what was actually deposited in the protein data bank. And it had iron, and zinc in this active site. We found that it was actually cobalt, manganese, and manganese. Now, it's important to say that PIXI cannot place these metals in the right place. All we can say is that, yes, you've got cobalt and manganese in here, but there's very little iron. But what we used was the anomalous data deposited in the protein data bank and used the energy that the data had been collected at, worked out what the theoretical anomalous signal should be, and then managed to match up the anomalous peaks with these metals and actually locate them all. So when we did that, this chain A, there's 3 chains, A, B, and C. Chain A turned out to have a sulfur, an SO4 actually bound here, and we realized that this protein not only is a phosphatase, but it's also a sulfatase. So the map was a real wasn't very nice here, and these metals were the wrong ones, and they hadn't identified that there was a ligand in there. We've published this. Chain B was a similar mess, but 2 of them were correct. They got 2 ions and a zinc, but it was actually cobalt and 2 ions. And then chain C, they had iron, and zinc in the protein data bank, and cobalt, iron, and manganese. So this was a complicated example which we were only able to untangle because of the anomalous signal. And I'll just show here how we did that with the anomalous. I won't go into it now, but basically we were able to place the metals, whereas the Pixie technique on its own can't, but in combination with the anomalous signal it can. And that was our paper, which came out in JACS. And I'm very happy to say that the original authors of the first set that we did, the first 32, which was John Hunt and Gaetano Mataloni and Liang Tong, they all agreed to be authors on the paper because this was extra information. It wasn't that they'd made a mistake, it's that they'd done a reasonable job, but then it needed extra information to tie it down. And this is Jeff Grime here, who I mentioned already, and the student who worked on this, Oliver Zeldin, who sadly now works for Facebook, and Eddie Snell, our collaborator who got hold of the, who had the proteins, and Elizabeth Snell, who did some of the refinements, a Part 2 student who's now an advertising executive, David Yates, and an undergraduate master's student, Peter Woodcock, who did a lot of this work too. These are all my funders over the years, so thank you very much to them all. And this is was my group in 2021, I think, from all over the world. Been fantastic fun working with them all on all these problems. And this is where they are now. As you can see, they're in Spotify and all sorts of Siento, cybersecurity and so on. So we're feeding all sorts of industries here and my collaborators. And thank you very much for your attention. I'd urge you, if you have anything biological, think about publishing in our structural biology Acta Crystallographica D, that I'm an editor there, and there's Acta F for papers without which haven't got as much information, and I'd be happy to take any questions.
Thank you very much, Elspeth. So please thank Elspeth for a nice talk. Yeah, physically.
Oh, hello, Spicek.
Yes, unmute yourself, please.
Great to see you.
It's my pleasure. I have one remark, maybe 2.
Yeah.